Hey everyone, welcome back. We're going to talk about osmosis in this video, and I'm going to just use purple as my color for osmosis for no reason other than it just sounds fun. So, the definition of osmosis, this is the diffusion of water. So, in the past, or in the last video, we looked at diffusion of the solute. Now we're looking at the diffusion of the solvent or water, diffusion of water. Down its concentration gradient, okay? So from higher to lower concentration. And this again takes no energy. So we're moving down the concentration gradient just like in the diffusion, but now we're moving water molecules. So water can pass through either directly across the plasma membrane. Water molecules are polar, but these are special, okay? There's always something that breaks the rules. Well, water is what breaks the rules in diffusion. Water is the only polar molecule that can actually pass through the plasma membrane. And so it can move across the plasma membrane directly, but it typically uses these special channel pores. So these channel pores are called aquaporins. Aquaporins are specialized pores that are built into the plasma membrane to allow a lot of water to move in. All cells need water, so cells can't survive if they dehydrate in general. And so what happens is when dehydration is occurring, the cells will start producing more aquaporins to pull in more water. And water moves just like any molecules in with diffusion. They're going to move in both directions, but they're going to move in a faster direction down their concentration gradient than against that gradient. So let's look at osmosis. So here we have a beaker of water, or a beaker of a solution. Within that solution, we have a 70% water to 30% solute ratio. Then we have another container. Think of this as maybe a balloon of some sort or something that has a selectively permeable membrane on it. So inside of this container is a solution containing only 40% water and 60% solute. So there's more water in the solution than in the container. So what happens is water is going to move down its concentration gradient from higher to lower concentration into that container. Okay, and so it's going to keep moving. Now, there's always movement both ways, so water is also going to move backwards, but it's going to move faster into this container until we have equal amounts of water to solute ratio on both sides. So there are three types of water or solutions that we can talk about. There are solutions known as isotonic solutions. So when I think of an isotonic solution, I always draw a beaker, and my beakers are actually just little squares, and I draw a cell, okay? So in the beaker, we'll put a 40% solute, which means we have a 60% water ratio, okay? So in the cell, if this is isotonic, we also have a 40% solute and a 60% water ratio. So we have equal water and solute on both sides. So what happens is water is going to move into the cell and out of the cell in equal amounts. So we have movement in and out all the time, but there's no change in the amount of water in or out of the cell. So we can draw that same exact setup here. So we still have the solution and the cell. So let's use the exact same cell. So we have a 40% solute and a 60% water, okay? So 40% solute, 60% water. I didn't put the percent sign there, but you see it there. And in a hypotonic solution, the solution has less solute than the cell. So that means the cell has 40% solute, so we'll give the solution, it has less, a 30% solute. And that means there's 70% water, right? So now we have 70% water here, 60% water there. So what happens is water's going to move faster into the cell and slower out of the cell. And so this is a hypotonic solution. It causes cells to swell up. And I always remember this because hypotonic sounds like hippo, right? Hypo and hippo. Hippo. And hippos are large. So cells get large, they swell up, and they can actually burst if they are not, if you're not careful. So then in the last solution, this is a hypertonic solution. We use the exact same cell again. So we have 40% solute, 60% water. And the solution in this case, the solution has a higher solute ratio than the cell. So it's the exact opposite of this one. So now we're going to make this, say, a 50% solute and 50% water, okay? So the solution has 50-50 solute to water ratio. The cell has a 40-60 solute to water ratio. So now there's more water in the cell. So what happens is more water leaves the cell than enters the cell. And so we end up having the cells shrink. And so they shrink because they are dehydrating. And so in both cases, hypertonic and hypotonic solutions, you can kill a cell, whereas isotonic solutions maintain the health of a cell. So here we're looking at animal cells. In this case, it's actually blood, okay? So here we have red blood cells, and in an isotonic solution, you have water moving in and out at equilibrium. So there's no net movement of water in either direction. So the cell stays healthy. Here, in a hypotonic solution, we're going to have more water moving in than moving out. And so the cell's going to gain water, it's going to get larger, and the cell ends up potentially bursting. And in this last solution, you're looking at the same blood cell, but in this case, it's in a hypertonic solution. So there's more water leaving the cell than entering the cell. So the cell loses water, that's dehydration, and it causes it to shrivel up. And when cells shrivel up, that is known as crenation. So when a cell is in a hypotonic solution, it can burst, and that would be called cytolysis. Whereas if the cell is in a hypertonic solution, that cell is going to crenate. In plant cells, the same process can occur, but because plants have a cell wall, that cell wall protects them. So when we have a lot of water moving in a hypotonic solution, That causes the cell wall actually increases the pressure, so you have an increase of pressure in the cell, but that cell wall keeps the plant from bursting, or those cells from bursting. And that actually is known as turgor pressure, and it actually is what causes plants to be able to stay upright. But in plants, in hypertonic solutions, they're still going to dehydrate because the water is going to leave, and the cytoplasm actually moves away from the cell wall, and that's called plasmolysis. So we'll stop here and we'll get into facilitative diffusion in our next video. Bye.